In 1999, a group of hunters found a mummified body in Ts'ìnchi Arctic Park, which is located at the north of British Columbia. This mummified body was named Kwäday Dän Ts'ìnchi, or more commonly called the Canadian Iceman. By using radiocarbon dating, it shows the Iceman was born during 1450 to 1700 AC. Dr. Monsalve from University of British Columbia was participating in the research of tracing the origins of the Iceman.
One day, my husband woke me up and told me, "Vicky, they just found a mummy frozen in a glacier in British Columbia." And I said, "Oh, this is a nice trick for him to wake me up." You know? So, in fact, I went later on to read the newspaper, and this was true.
We all know that cell is the basic unit of living organisms. We can find DNA in two places within cells, nucleus and mitochondria. Mitochondria produces DNA independently from the cell. Mitochondrial DNA are passed through mother's eggs directly to sons and daughters. In turn, daughter passes the same mitochondrial DNA to her offsprings. This is a great source to trace back the origin of the Iceman. In Dr. Monsalve's research, this technique was heavily used.
Because environmental factors like UV radiation, humidity, or heat can degrade it and decompose the DNA of organisms that are dead. So the probability of getting different pieces, small pieces of this, the likelihood is higher than getting nuclear DNA.
The quality of the sample is determined by the macromolecular analysis. This analysis measures the extent of preservation by looking at the carbon and nitrogen content preserved in the body.
The purpose for using studies of proteinaceous material is to see the likelihood of getting DNA. So if you have a good percentage that is normally comparable of normal tissues, the probability of getting suitable DNA for analysis is very high.
PCR is a well-known technique in molecular biology. It functions as an amplifier of a segment of DNA by starting from one copy of few of DNA segments. After a number of rounds of PCR, the DNA segments get duplicated to millions. As the sample resources were really limited, Dr. Monsalve applied this technique.
Normally, the cycles that for contemporary tissues is 30 cycles. We increased to 40 cycles. But it's the same procedure.
Back then, the research time was quite limited. Dr. Monsalve and her team, however, still got some interesting findings.
Initially, we found that one individual Haida had the same mitochondrial DNA sequence, and individuals for Central America also had the same mitochondrial lineages, and from South America, 3 individuals from Brazil, probably mixed with the indigenous genes. No?
Later on, a further DNA testing of more than 240 volunteers from the local First Nations revealed 17 persons who are related to the Iceman. Please check more details for interesting findings and results in the podcast.